And now, I'd like to give the floor to Maria Poptsova, High School of Economics, School of Informatics, School of Computing Science. Dear colleagues, hello. My presentation, I'd like to begin with an introduction on the trends of technical progress development with the words of Ray Kurzweil, the technical director of Google, who is quite famous for his precise predictions. Beginning with 1990, he had been predicting the development of biotech and has made 147 predictions at 86% accuracy using the neural machine learning terminology. He also published a book, Dawn of Singularity, with specific predictions year by year. If we predict the development, we will see that first decade will not see any transformation of biology or healthcare. I work at the biocomputer science lab, and we're into all of these multiple research vectors. We see self-driving cars, language translation machines, computers will grow in power, and according to his opinion, let's try and believe him, this singularity, we're not talking about progress yet, however, the singularity, the understanding of the workings of the human brain will fall at the mid-30s in this century. Around the year 2035, this accumulated data will produce a new technological breakthrough, and $1,000 personal PC will be 1,000 times more powerful compared to human brain. So specifically, looking at the years, at the predictions year by year, times of singularity in biology, humankind will be able to program cells, to reprogram cells, to cure diseases. This is certainly related with a giant leap in the workings of the brain, certainly. Now, my lab is focused on understanding these algorithms encoded in the genome. So let's take a journey into the development of genetic technology. It is quite an impressive journey, starting with the discovery of DNA in 1869 to the emergence of sequinators, third-generation sequinators, Oxford Nanopore technologies, or nanopore sequencing. So less than 150 years is all it had taken. If we look at the sequence, DNA sequencing, and the cost per megabase, we'll see that the speed of biotech progress development sequence here is not following the Moore's law of development beginning with 2007. At the slide, we see the curve exponentially fall, and now
in the United States, $100 per genome is the relevant pricing now, and actually additional technologies are around. Apart from just sequencing the genome, we will also have a number of similarly useful details on the structuring of information in the genome through the same technologies of sequencing that are accumulated, successfully advancing exponentially into the big data of molecular biology that in the format of cards is shown at the bottom. The whole of epigenetics and transcription factors, and of course, without machine learning, Human brain cannot digest the whole thing. If we sum it up with the medical data, then for the coming decades, it means that we will have a lot of tasks for the data sciences. And as part of our applied activities, we selected cardiogenetics, and the Higher School of Economics was the initiator of setting up a consortium for epigenetics for cardiovascular diseases. Actually, the genetics of such diseases is an area that lags behind in terms of its development from cancer genetics across the world, and only in Russia, because probably it receives less attention and is not offering quick results. But this is a very relevant subject, and we initiate a lot of pilot projects. I'm going to speak about one of them. This is a very powerful and relevant area now. Pharmacogenomics, how genetic conditions impact the effects and side effects from taking medical drugs. And this project is done in cooperation with the Russian Medical Academy of Continuous Professional Education, and both the academy and the project is led by Dmitry Sychov, professor, corresponding member of the Russian Academy of Sciences. We started this cooperation before COVID. The task was to use machine learning systems to predict adversarial impact from anticoagulants and antidepressant drugs. So this is the area that is called machine learning for personalized medicine. So what is anticoagulant all about? You see a very beautiful photo of a dangerous thing, thromb. Fighting against thrombosis was always relevant, and these medicines were prescribed for uh, and also deep vein thrombosis, heart fibrillation. Also, over the recent period of time, these medicines are prescribed to patients with COVID-19. And this can also be seen through growth of consumption. To the left, you see a chart showing the growth of the market, sales market over the recent year. This is the dynamics of anticoagulant sales. You see the spike from 2019, 2020, and this shows various types of drugs. And simultaneously with that, mortality also grew from bleeding in gastrointestinal area, and even non-experts understand why, because these drugs are taken in the wrong way and the doses are wrong. And if we look at another piece of statistics, we can see that at the same time, if we just look at the reasons, then individual perception of drugs, mistakes in personalized medicine is 65%, as the chart shows, and 35% is probably deviation from clinical prescriptions or lacks and drawbacks in organization of treatment in a hospital. And here we show an example of our joint work before COVID, where through methodologies of machine learning we try to predict mortality for patients with triple therapy with these three medicines: aspirin, clopidogrel, and rivaroxaban. And we see that machine learning approaches work quite effectively, and they do almost no mistakes in predictions for mortality. And another area is ethnic area related to the fact that there are markers that are associated with the effects of anticoagulants, and the frequency of these alleles are different in various ethnic groups. So there's differences between ethnic groups in sensitivity towards clopidogrel. We've described it and published the material together with a group led by Dmitry Alexeyevich. And just to show you why it happens, this is a protein coded in the gene cytochrome, and its polymorphism is associated with reduction of the effect of this anticoagulant. One mutation in this gene, one spot change actually makes the activities of this drug ineffective. And the impact will be different depending on the population. For Europeans, it is enough to have a mutation 1 allele, but for Asian people too. So in Dmitry Sichov's group, the work is done to prepare an atlas of frequency of alleles across the whole of Russia. So they take a biomarker and genotyping the whole population of Russia. You see how the frequencies are beautifully shown on the map of Russia. Different colors mean different frequency of distribution of the specific allele for susceptibility towards an anticoagulant, and it means different dosage. And the dosage, graphene is an old one, but the dosage is different by half a pill for Yakutia, for instance, and for other areas of Russian Federation. And the same research was done for another gene that is associated with the effect of another, a more modern medicine. In our work, one of the tasks to implement into clinical practice is to prepare a formula for calculation, considering pharmacogenetic specificities of various ethnic groups. So we tried through Poisson regression, we tried to show such complicated formulas. This might be efficient not for dosage, but for clinical indicators. So this is the indicator. PRU is an indicator you can clinically measure to understand how much anticoagulant, how it works, is it effective or not. And the tasks that we're facing in the future, considering ethnic specificities of the population and all omics data that can be accumulated for patients and that will be accumulated now through such data, we plan to predict the level of risk on tolerance towards such anticoagulants and proper dosage prescriptions for the population for a specific ethnic group. Thank you very much. That's it from me. Thank you very much, Maria. I have a couple of questions I will read from our chat. A question related to applying such genetic data. Is it safe to go through tests for sequencing? Can people who have this information use it against me? Probably it is relevant for any technology in AI, but anyway, the legislation says that the data for analysis is going in an anonymous way. When databases are analyzed, information on owners of such genomes is cleaned up. There is no danger in this. But how to offer treatment efficiently and how to structure personalized treatment, this is a very important point for such cases. And another question, at the beginning of your presentation, you mentioned multi-bundle data when you have a genome and clinical case. For instance, countries like England has large databases with such content. But could you please tell us such databases, when will they come to Russia? What are the prospects? Are there any prospects? Yes, there are, sure. Now, the Russian genome project started. It'll be a KBA bank, 100,000 genomes, and we will have the same thing here in Russia. And every hospital, every medical organization is now accumulating data on patients, and everything is being digitalized now in an exponential way. It'll be developing, I think. And if we are working to improve systems that will be able to combine such data effectively, then I think, as Ray Kurzweil says, by 2030 they will become very powerful companies in analyzing heterogeneous data. Okay, understood. Thank you very much.